Welcome back to Dr. Liu's VCE Biology. Today's topic is DNA manipulation techniques and applications, as presented in the 2025 NHT Biology exam. Let's learn together. If teachers, parents, or students notice mistakes in my explanations, please provide me with your feedback. Type 1 diabetes mellitus, T1DM, can be caused by genetic mutations. DNA profiling was carried out, using gel electrophoresis, to identify if children carry the specific genetic mutation for T1DM. Children without T1DM have fragments of all the same length that run furthest from the well. Any other combinations of fragments result in children being diagnosed with T1DM. Each of the lanes 1 to 6 represents a different child tested for the genetic mutation. Lane M is the DNA base pair, BP, ladder. The results are shown below. In individuals without type 1 diabetes mellitus, T1DM, gel electrophoresis reveals DNA fragments of a uniform length that migrate the furthest from the well. The presence of any other fragment combination indicates a T1DM diagnosis. Based on the gel results, lanes 1 and 4 showed the furthest running, uniform fragment length, confirming that these individuals are healthy, non-diabetic. Conversely, the differing fragment patterns observed in lanes 2, 3, 5, and 6 lead to a T1DM diagnosis for the children represented by those lanes. To diagnose type 1 diabetes mellitus, T1DM, using a gel electrophoresis map, the target DNA is first amplified by polymerase chain reaction, PCR, to ensure an adequate starting amount. The amplified DNA is then cut into fragments using a specific endonuclease restriction enzyme before being separated and visualized on the gel. Cavendish bananas make up around 97% of bananas grown in Australia. The plants can be affected by a disease, tropical race 4, TR4, which causes the plant to wilt and die. The disease is caused by a fungus that remains in the soil for years and cannot be detected. The disease first appeared in 2015 in northern Queensland and is now found on 9 farms. The fungus is easily transmitted, and there is no treatment for this disease. Australian scientists have found a wild variety of the banana plant that is resistant to TR4 disease due to the RGA2 gene. Cavendish banana plants also have the RGA2 gene, but it is not functional. Both the wild variety and the Cavendish banana plants are different subspecies of Musa acuminata. To transform the Cavendish banana plasmids from bacteria were used. An image of plasmids within a bacterium is shown below. The functional RGA2 gene from the wild variety of banana was isolated and then incorporated into a bacterial plasmid. Cutting the plasmid, the endonuclease cuts the circular plasmid at a specific recognition site, opening the ring and producing sticky ends, short, single-stranded overhangs. Cutting the RGA2 gene, using the same enzyme on the RGA2 gene ensures that the ends of the RGA2 fragment have complementary sticky ends, meaning they match the sticky ends produced on the opened plasmid. Joining bonds, DNA ligase catalyzes the formation of the phosphodiester bonds that covalently link the sugar phosphate backbones of the RGA2 gene to the plasmid DNA. The functional RGA2 genes were transferred into the genomes of Cavendish banana cells using the plasmid. These banana cells then grew into Cavendish banana plants resistant to TR4 disease. The TR4-resistant Cavendish bananas are approved for food production in Australia. The bananas are considered safe and nutritious to eat. However, There are no immediate plans to grow the TR4-resistant Cavendish bananas on a large scale or sell them to consumers. The TR4-resistant Cavendish banana is considered genetically modified, GM, because the RGA2 gene came from the same species, M. acuminata. However, it is not transgenic because, by definition, a transgenic organism has had genes inserted from a different species. Despite being approved for food production in Australia and deemed safe and nutritious to eat, there are no immediate plans to grow or sell the TR4-resistant Cavendish bananas on a large scale. Economic and market. The primary resistance is economic, as Cavendish bananas are not currently in short supply in Australia, with only 9 farms infected with TR4. This lack of market urgency, combined with the cost and time required to upscale research and cultivation of the new variety, reduces the incentive for immediate large-scale production. Legal and social hurdles. Adoption faces significant legal and social obstacles. The TR4-resistant plants must be labeled as genetically modified, GM, a legal requirement that creates a commercial risk. This GM labeling can deter consumers and reduce the guarantee of purchase. Overcoming this resistance would require extensive education and marketing campaigns for both farmers and consumers. Techniques: 5 marks, T1DM Diagnosis, Q7, and Q8 T1DM mutation is identified on a gel map by fragment bands that are not the furthest running uniform length, which indicates a healthy individual. The process involves PCR amplification, cutting the DNA with an endonuclease, and then running the fragments through gel electrophoresis for visualization and analysis. Recombinant plasmid, Q3A. A recombinant plasmid is formed by cutting both the gene, e.g., RGA2, and the plasmid with the same endonuclease to create complementary sticky ends. The fragments are then joined together permanently using DNA ligase. Implications: 5 marks. Classification: Q3 by the TR4-resistant Cavendish is a genetically modified, GM, organism, but not transgenic. It is GM because the RGA2 gene was inserted, but it is not transgenic because the gene came from the same species, Musa acuminata. Adoption hurdles: Q3b. Despite being safe, the GM banana faces low adoption due to a lack of economic urgency, current supply is stable, and significant social risk. The required GM labeling deters consumers, indicating that substantial education and marketing efforts are needed to justify the high cost of large-scale production. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.